Assalamualaikum warahmatullahi wabarakatuh, and hi everyone! So today our group will be presenting about determination of natural pigment in fruits by using paper chromatography. So first of all, let me introduce our team members. My name is Amira and my team members are Anisha Hira, Nurfatia Sofia, Fatim Farzana and Nur Afifa. Now let's move on to our introduction. So what is chromatography? Chromatography is an analytical technique used to separate a mixture of chemical substances into its individual components. This gives us the opportunity to truly analyze each component present in a mixture. There are several types of chromatography, including gas chromatography, high-performance liquid chromatography, thin layer chromatography, and also paper chromatography. So what technique we use in this experiment again? Yes, it's paper chromatography. So what is paper chromatography? Paper chromatography is an analytical technique used to separate and identify dissolved chemical substances by taking advantage of their different rate of migration across a sheet of paper. So why does we choose this technique? Well, first, because it's simple and low cost. It's beginner friendly because it does not require any advanced instrument, neither does complex procedure. It's also budget friendly due to the uses of cheap and widely available materials such as ethanol and distilled water. It's also time effectiveness because it can provide fast results that can be observed within the first minutes. It makes it suitable for classroom experiment and also
There are two objectives in this experiment. First, to identify the pigment presence in fruit extract by using paper chromatography. Second, to separate the pigment in fruit extract based on their solubility and the interaction with mobile and stationary phase. Now, I will explain to you some natural pigments present in fruit. First, we have anthocyanins. Anthocyanins are pigments that found in many fruits and vegetables, bringing out the red, blue, and purple colors in them. Anthocyanins are part of polyphenol group and they dissolve easily in water, which means it water soluble. These pigments are pH dependent. Besides add color, anthocyanins also act as a powerful antioxidant that can help to protect our body from harmful molecules. They provide various health benefits such as reducing bad cholesterol, which is LDL, and lowering blood pressure. Next, we have chlorophyll. Chlorophyll is the green pigment that gives plants and unripe fruit their color. As we all know, chlorophyll plays a crucial role in photosynthesis, the process where the light is turned to energy for the plant. In most fruit, chlorophyll will break down as they ripen, causing a change in color. However, in some fruits such as avocado and green apple, the pigment stays even when fully ripe because they retain their chlorophyll. Chlorophyll is fat soluble. Chlorophyll also provides benefits for human. For example, it acts as a natural detoxifier, support liver health, and even help to maintain healthy skin. Last but not least, we have carotenoids. Carotenoids are natural pigments found in fruit that give them their vibrant yellow, orange, and red colors. All carotenoids share the similar molecular structure and are classified as tetraterpenoids. Carotenoids are divided into two groups, which are carotenes, where they only contain carbon and hydrogen atom, and xanthophyll, where they contain oxygen atom. Carotenoids are fat soluble, meaning they do not dissolve in water, but dissolve in oil or alcohol. Besides giving color, carotenoids also provide various health benefits for humans. For example, some carotenoids help to maintain healthy vision and may reduce the risk of certain types of cancer by inhibiting the growth of abnormal cells. These are the fruit samples that we will be using in this experiment. We have strawberry, cranberry, and blueberry.
Next is pigment composition. Do you know that capillary reaction process involved in paper chromatography? Capillary reaction is the driving force that allows a liquid solvent to move up a piece of chromatography paper. Different pigments will travel at different speed due to their affinity, solubility, and polarity. Affinity is how strong the substance sticks to the surface. If a pigment has high affinity to the paper, it will stick more and move less with the solvent. The second one is solubility. Solubility means how well a substance can dissolve in a liquid. Pigments that are more soluble in the solvent will move faster and further up the paper. For example, pigments that dissolve well in alcohol like anthocyanins will travel far than those that don't. The last one is polarity, refers to the distribution of electrical charge in a molecule. Pigments from fruit can also be polar and nonpolar. Polar compounds may stick to the paper more, while nonpolar compounds travel farther in a polar solvent. For example, carotenoid is nonpolar, which travel farther in a nonpolar solvent. Meanwhile, anthocyanin is polar and travel less in nonpolar solvent. Now, let's move on to the procedure of paper chromatography. First, choose fruit that are rich in pigments from anthocyanins like strawberry, cranberry, and blueberry. Secondly, the fruits are crushed using a mortar and a pestle. Then, filter the crushed fruit through a cheesecloth to collect the juice. Third, cut a strip of filter paper about 4 cm wide and long enough to hang into a beaker. Draw an original line lightly 2 cm from the bottom using a pencil. Fourth, place a small drop of the fruit extract on the original line using a capillary dropper.
For step 5, add 70 ml of ethanol, 30 ml of distilled water and 1 to 2 ml of white vinegar into a beaker and mix it together to create a solvent solution. And then, pour around 1 cm of the solution into a new beaker. For step 6, hang the paper strip into the beaker and make sure the pigmentous part didn't reach the solvent solution. And make sure to wrap the beaker with a cling wrap to avoid evaporation. Now, let's go through the continuation of the procedure. For step 7, as the solvent moves up the paper by capillary action, the pigment from each fruit will separate into different color bands depending on how fast they travel. The diagram shown here is just as a sample. The actual result may vary. For step 8, once the solvent front is around 2 cm from the top of the paper, remove it from the beaker immediately. Before it dries, use a pencil to mark the position of the solvent front. Lastly, repeat step 6 to 8 with the remaining fruit extract using the same solvent. Then, observe the difference in pigment separation between each fruit. Here are some example results from the chromatography experiment. As shown, each fruit extract produces different pigment bands. The strawberry extract shows a strong red pigment. The cranberry extract has several different bands ranging from pink to dark purple. And the blueberry extract displays shades of blue and violet. These separated bands represent different pigments present in each fruit, including anthocyanin. Now, let's look at the RF value obtained from the chromatography. RF stands for retention factor, and it tells us how far a pigment travels compared to the solvent. It is calculated using the formula RF equals the distance traveled by solution divided by the distance traveled by the solvent. For example, the strawberry pigment traveled 1.8 cm while the solvent front traveled 6.0 cm, giving an RF value of 0.30. Cranberry showed an RF of 0.33 and the blueberry extract had an RF of 0.50. Since the RF range for anthocyanin pigment is between 0.30 and 0.62, all these results confirm the presence of anthocyanins in the fruit extract.
In conclusion, this paper chromatography experiment showed that it is a simple, low-cost and quick method to separate and identify natural pigments in fruit. We were able to observe pigments like anthocyanins, chlorophyll, and carotenoids, each moving differently based on how they dissolve in solvent. These pigments not only give fruits their color but also provide important health benefits such as protecting the body, supporting eye and skin health, and lowering bad cholesterol. Overall, the experiment helped us understand both the techniques of chromatography and the importance of natural pigments in our daily life. That's all for our presentation. Thank you for listening.